welcome to our afternoon session. Let's get started so we can keep on schedule because I know the fireside chat people don't want us to use up their time. So I'm going to try to keep us going on schedule. But hello, welcome everyone. I am Colleen Lynch. I am the Senior Curator of Animal Care here. I made this word cloud to try to talk about all the things that we do here at SCBI, and it turned out really, really busy because in fact, we're really, really busy people. We're doing lots and lots of projects with lots and lots of partners on lots and lots of species. And we want to give you a glimpse into that world today. But no matter what we're doing on all of those projects, we really look at excellence in animal care as our foundation, that our animal care is informed by science and it informs science. And everything we do is based on that foundation, whether it's for individual animals, for species, for conservation in general. And that's really made us leaders even beyond our own perimeter fence and beyond our own borders here. So with all of the alumni in the room, I do want to take a moment to recognize the alumni who really influenced me in my career. And thank you all. These leaders helped to bring me here. And I know that you all of leaders have helped and supported many other individuals like me. Thank you so much for that contribution. And now I want to let you meet the leaders of today, the leaders of tomorrow, and share their stories with you about animal care here at SCBI. So thank you.
Good afternoon. My name is Dolores Reed, and I have been here since 1986 and have been the ungulate or hoofstock curator for the last 10 years. I'm going to talk a little bit about some of the species that we've housed here at SCBI for the last 50 years, focusing mostly on hoofstock. Kevin already mentioned some of them. There have been 19 species of ungulates that have come through the center. It's actually 20. I just found out yesterday that I skipped one. And we're using 7 barn complexes. 3 of those photos are shown on the right there. All 3 of those have what we call hydraulic restraint systems in them. And the photo at the bottom shows one of the hydraulic restraints, which gently squeezes the animal, picks it up, and allows us to do ultrasounds, injections, many veterinary procedures that we might need. Now, the first species that came into SCBI arrived 51 years ago, actually, in June of 1974. That was the scimitar-horned oryx. 2 females, one named Louise, the other one named Gil, arrived, and both of them thrived and survived well into the 1990s. Some of the research carried out with the oryx consisted of testing the GPS collars for fit and reliability, as well as behavioral studies. This research contributed to the release of the oryx back into the wild in Chad that Catherine had talked about this morning. The success of the release program, as she said, downgraded the oryx on the IUCN Red List from extinct in the wild to endangered. That was quite an accomplishment for us to be part of that. The next species that arrived at SCBI was the Bactrian camel. They arrived in December of 1974, and I don't know too much about them. I do recall hearing stories from other keepers about their antics. One can still see the bite marks of these guys on the mead barn rafters. Our next species is the Burchells, or one of the plains zebras. They were a test species brought in to see how well they might handle Virginia's climate. They did quite well. They were definitely a talking point for anyone driving down 522 that may have caught a glimpse of them outside of Gate 6 on the Mead hillsides. Elsdeer is a species very near and dear to me, no pun intended. They are a deer species. I have been the North American population manager and stud bookkeeper of this species for over 10 years and have worked with them directly for over 25 years. Managing such a fractious species has been a challenge. It has been very rewarding to gain their trust and ultimately to be able to transfer what we have learned in their management back into facilities in Thailand, where research on the species has continued, resulting in successful embryo transfer. The next species is the Pierre David deer. This was the largest number of individuals of any ungulate species that SCBI has ever held. Over 100 individuals were held in 3 herds up into the late 1990s. These herds were a great resource for the training of scientists in field research, such as how to create transects to estimate wild population sizes and densities in the wild. Next, we have Reeves muntjac. This was the first small cervid held on site. They did quite well despite their small size, and their size required some more intense predator prevention management development. Next, we have the Persian onager. They were actually accessioned in 1977, left for a while in 97, and returned back in 2008. They are unique in in this perspective that they have come back. Usually when a species leaves, it hasn't come back. There are currently less than 40 onagers genetically managed in 3 North American facilities. This is one of the species we currently manage using a hydraulic restraint system, as seen in the photo at the bottom. Sable antelope, one of my favorites, tend to be hard to manage in zoos due to their very aggressive temperament. They did very well in the big open pastures on site. Along with the scimitar horned oryx, sable were a pioneer species managed in a drop chute system, which was a precursor to our current hydraulic restraint systems. The drop chute consisted of a ramp that went up to a V-shaped platform with a flat bottom. The floor, once the animal was inside, the floor of the V dropped out, and it dropped the animal into the V, wedging it, and the animal we could handle and draw blood, whatever we needed to do while they were in the wedge, and then the sides released and released the animal. Przewalski's horses. This is a photo of Anne and her daughter Christine, who is 1 day old in this photo. In 2013, Christine was the first ever Przewalski's horse, or P-horse, foal produced by AI. SCBI pioneered the hydraulic restraint design and animal handling systems we use today with this species, including teaching the mares to urinate on command to facilitate hormone monitoring for pregnancy. Yes, it is possible to teach a horse to pee on command. The Arabian oryx was another species brought in that was similar to the scimitar, but they were hardier and tougher than the scimitar. The females were quite impressive with the lengths their aggression could go to during neonate exams and treatments of their fawns. I'm sorry, not fawns. I should have said calves. They're in the bovid family, calves. Tufted deer is another small cervid species that we have here. The large photo is an excellent example of a male with his canine teeth visible just at the bottom of his lip there. The fawns, as seen in the small photo on the left there, they usually weigh about 2.5 pounds or 2.5 kilograms, 5 pounds, and have a secret weapon for predators when handled as infants. They can let out a blood-curdling scream when grabbed. Have you ever heard a rabbit scream? That's what these guys sound like. Dama gazelle. Dama gazelle is the largest gazelle species. There are less than 100 left in the wild. As Catherine mentioned this morning, they're also part of the release program that the oryxes are going through in Chad. SCBI has been involved in the testing of the horn-mounted GPS transmitters for use for the release along with the oryx. Dama calves, in the smaller picture there, are quite odd-looking with huge ears, and they remind me of Dobby the house elf from Harry Potter. They had a knack for pinning their ears back when they didn't like something, wrinkling their nose. They just looked really mad. And our final species, the most recent one that we got in, is the Hartman's mountain zebra. They are currently in training, familiarizing them with the hydraulic restraint systems at Green Hill Barn. The foal in this photo was born on June 26th of this year, and her name is Zara. Now, I don't have time to go into all the other hoofstock species we've had here, but here are a few of them. We had bongo for a short time, dik-tik, which is another small cervid, takin, we held a few. The bison, we had both bison and European weasant for some time, and pudu, another small cervid. Grebe zebra were here for a short time, and I don't have a photo of it, and the one species I missed earlier from yesterday is reindeer. We had reindeer here from about 1981 to 1985. A few other historical mammal species we have here you might not be aware of are the Matschie's tree kangaroo, golden lion tamarins, which you'll hear a little bit more about in a few minutes, Geldes, marmosets, or calamicos, tarsiers, and tiger quolls. We had one of the first breeding colonies of tiger quolls outside of Australia housed here at SCBI. Bush dogs, binturongs, and you might wonder, why did I say European polecats? They were very important to the black-footed ferret program that we have now. We had over 128 individuals here between births and accessions, and we had them here. You ask why? They were brought as a model species for the release program to learn how to breed ferrets and attempt to teach the ferrets to recognize and evade a predator. And down there I have listed Robo Badger. Robo Badger was a stuffed badger that was put on top of a one of those remote control cars to see if we could get the ferrets to be afraid of it. I don't believe it worked out very well. Paul would know better. He could answer that one later. With the space, facilities, and all we have learned in the management of ungulates and other species here, the future is limitless. Thank you for your time.
Good afternoon, everybody. I'm Adrienne Crosier. I'm Carnivore Curator here in Front Royal, and I'm joined today by Amber Dedrick, who is one of our carnivore keepers. So we're going to tell you all a little bit about our cheetah program, but first, a little history. So shortly after the foundation of this research facility in Front Royal, SCBI scientists started studying cheetah physiology. Our scientists were the very first to document reproductive metrics in the species, including the very high incidence of abnormal sperm in males. Working with geneticists, our scientists were the first to document the very low level of gene diversity in the species. And one of our scientists, Dr. Jo Gail Howard, was the very first to develop artificial insemination and produce cubs. Our decades of research have resulted in many projects and supported many students and have produced over 75 cheetah-specific peer-reviewed publications. So our legacy continues. In 2012, we were critical in the formation of the Cheetah Breeding Centers Coalition, or BCC. The BCC was formed for several reasons. One, to produce cheetah cubs, but also to more consistently produce animals on an annual basis for the species survival plan. We also wanted to just increase communication across the breeding centers and increase research collaboration. The BCC is very committed to husbandry training, and we hold training courses every 2 years to train the next generation on best practices in cheetah management. Our cheetah science facility here was established in 2007. It was obviously built to help produce cheetah cubs for the SSP on an annual basis and to study cheetah breeding management, but also to help promote our research projects and increase that research collaboration across the centers, and to help train staff on the art of cheetah breeding, because it is very much an art. This is an aerial of our facility. Our male cheetahs are kept at one end and visually isolated from all of our females and family groups, which makes a big difference when you're trying to breed. So cheetah breeding is a very complex and stepwise process. We physically move the males out of their enclosures and put them into a female's enclosure. They are then allowed to investigate her environment. They smell where she's been sleeping, defecating, and if they detect that she is an estrus based on smells, they will make a vocalization. That is a very happy noise for us. We love when we hear the males make this vocalization. That means they are sexually interested in the female that they've just been investigating, but they haven't seen her yet. So the next step is a fence line introduction. And if all signs are positive and we see things from the female, such as rolling on the ground or rubbing on objects, we will then move to a full introduction where hopefully we will have mating and 3 months later, cheetah cubs produced.
So how successful is SCBI at breeding cheetahs? We have had 19 litters born here since the opening of our breeding center in 2007, which averages to be about 1 litter per year, totaling 81 cubs born and 62 living to adulthood, which is a very good success rate. So another part of the breeding process is that we do work with the associations of zoos and aquariums to develop new breeding recommendations each year. Then, based on these recommendations, we can target certain male and female pairs. And here at the bottom, you can see some keepers moving a male to go investigate a female yard down Lover's Lane. And then go through the breeding process as Adrienne described. So our data that we've collected over the years indicates that we have done 1,785 yard investigations. And you can see that it's a kind of cascading number. So they do a lot of yard investigations, but they're not going to be interested in a female every time. So only when they're interested do they get to move on to the next step, which would be a fence line introduction, and so on and so forth. So over the years, though we've done that many investigations, we've had 66 breedings that have resulted in these 19 litters. And it is a time-intensive process when each investigation takes a minimum of 30 minutes. Keeper staff have spent around 900 hours working to breed cheetahs here. And it's important to note that since we have moved to this breeding center model of managing the cheetah population, we have seen an increase in the number of cubs born, and we are able to maintain a higher overall cheetah population across the SSP. So moving to this breeding center model of cheetah management has allowed for many positives in breeding cheetahs. One is that we have dedicated staff that know the animals very well and provide good continuity of care for these animals. We have a larger number of animals. We currently house 27 cheetahs right now, and that gives us a lot of mate options and a nice quiet environment. And these are some of our moms and litters over the years. And although this is a time-intensive and labor-intensive process, this work helps the cheetah SSP goal of maintaining a sustainable North American cheetah population well into the future. And we are excited to announce that litter number 20 is on the way and is due in October. Thank you.
All right, so my name is Erica. Good afternoon, everyone. I work here in Front Royal with our Pacific Island bird species, mainly with the Guam Sihek. And this species is one of the rarest one that we work with at both campuses. They are extinct in the wild, and there are only 130 animals in human care. So Smithsonian has been involved in this project since the very beginning in the 1980s when a team of biologists went to Guam to collect the last 29 individuals that were left in Guam. And they were brought into human care to start the recovery project. Now, some of the team of those biologists were Smithsonian curators and biologists that went to help collect them. And since that time, we've been working on breeding the species. So we've been hatching chicks since 1985. But past breeding and working on the husbandry, we've contributed to a lot of other aspects of the program. And this is not an exhaustive list, but many of the things on this list involved multiple Smithsonian departments across Smithsonian and the zoo. The most recent project we worked on with the Migratory Bird Center was a collaboration on a transmitter attachment study that would help us to prepare for wild releases. So the Migratory Bird Center helped us figure out what type of harness and what type of material would work best for this species, because as we found out, they are particularly good at removing transmitter harnesses. So we found one that would work and that would help us to prepare for release. Now, we can't yet release this species on Guam because the reason they are extinct in the wild is still a problem, the invasive brown tree snake. So the next best option was to do a translocation, or creating an experimental population in a place where they did not previously exist. And so that location was chosen as Palmyra Atoll. And so in 2022, I had the opportunity to go with a team to assess this habitat, look at food availability for Sihek, and make sure that we weren't introducing a competitor for other species, and looking at nest sites. And so it was deemed a good habitat for Sihek. The only thing about this habitat is it's highly protected. They have very, very strict biosecurity restrictions. And so we couldn't just show up with adult birds. So what we did was come up with a stringent biosecurity protocol. And we had zoos from all over the country send their eggs to one central location in Kansas. And so at this facility that I managed, we received eggs, we artificially incubated them there, hatched the chicks, hand-reared them until they were independent. And at that point, they were put in their mosquito-proof carriers, so still in quarantine. and flew them from Kansas all the way to Palmyra Atoll. At that point, they went into their acclimation aviaries, and we were looking for a variety of behavioral standards before they could be released. And so the main one of those was obviously needed to be, could they catch and dispatch their own live prey? Keeping in mind that they had never seen any of these prey items because of the quarantine situation. So they did exceptionally well going after hermit crabs, geckos, huntsman spiders, cockroaches, anything really that moved in their aviaries. So we were pretty pleased with that. And once they passed all of those standards, we did a thorough health screening. And then I was able to attach the transmitter tags that we had tested here in Front Royal. So we gave them a couple of days after that, after attachment, to get adjusted to the harnesses, preen their feathers around them, make sure they were comfortable. And at that point, they were ready for release. And keeping in mind, this species had not existed in the wild for 36 years at this point. So we had 9 birds to release last year, and we weren't sure what they would do after release. And to no one's surprise, many of them went and harassed a lot of the native birds that were 3 times their size. They're known to be territorial, but overall they did pretty well with adapting and finding food immediately after release. So with a translocation like this, we would expect maybe a 50% survival rate. So we held our breath in the days and weeks and months after that to see what our survival rate would look like and how well they would adapt. Would they get along with one another, and how would their territories work with one another? And so I'm excited to share that this past weekend was the 1-year anniversary of the release, and we have 100% survival. And so everyone is doing fantastic, and actually some of their recapture weights have been a little higher than we would expect. So they're doing quite well and eating all the things. They have even nested, laid eggs, and we expect chicks as early as next year. And so all of this data that we're collecting on a daily basis, observing the birds, will help us to conduct releases in the future on Guam so we can return them home.
Hi, everyone. I'm Brian Gratwicke. I am the amphibian conservation biologist here at the Smithsonian National Zoo and Conservation Biology Institute. I first arrived at the zoo on a cold winter's day in 2004, and I was rescued by the kind folks in Amazonia who said, here's a nice warm place where you can look at wildlife all day long. So I started out with Ed Smith as a volunteer. And I think that's where I first met Randy. So it's been a long time, but I really wanted to break into conservation. That was my objective for moving to Washington, D.C. So after about 5 years, I eventually did land a job at the National Zoo to lead the amphibian conservation efforts. And this program was set up in 2008, which was Year of the Frog. And AZA led a huge campaign to persuade other zoos to get on board and try and build an amphibian ark for the frogs. So I was hired. I was given 6 months of salary, a desk, and a computer, and told to go save the frogs. And if I was successful, then I could stay. So I'm pleased to report that I am still here. And our most recent project was here in Panama in the Campana National Park. It's Panama's first national park that was created. Beautiful montane habitat. It is also the home of the Panamanian golden frog. But we have done an outrageous thing. We attempted to reintroduce 4 species of frogs at the same time. So the first idea was to see if we could restore a population of Pratt's poison dart frogs that had gone extinct from this national park. They had disappeared. Our project director, Roberto Ibanez, had studied this frog for his PhD, and he had studied the calls of this frog in Campana National Park. And we spent 2 years looking for them and using passive acoustic monitoring and AI to see if we could to relocate this frog, and we couldn't. But we had found a population somewhere else where the frogs had recovered. This is Bajo Bonito, and we got with our partners at the Cheyenne Mountain Zoo in Zoo, New England, and we made a little expedition out there, and we managed to collect 12 pairs of animals, male-female pairs, and we moved them to, do we get sound here? But we'll try that again. So we moved them to Campana National Park, and you can hear the sound of Campana National Park restored. And it was a really magical moment. These frogs were kept in these kitty play tents for 2 weeks, and then they were released. And at the time that we were, here's a frog calling on a rock inside the kitty play tent. They rather liked their digs. And if you look carefully behind this female frog, she's sitting on a clutch of eggs that was laid inside the kitty play tent in the 2 weeks that we were just acclimating it to the new environment. And she took the tadpoles onto her back. And when we opened the kitty play tent, she went off and took her new clutch of eggs into the wild. So that's probably got to be the fastest reproduction following a translocation that we've seen. We also tried a pilot release of crown tree frogs. This species was last seen in the wild 15 years ago in Panama. And so we have these breeding in captivity. They're doing pretty well in captivity. And we have worked with our Reptile Discovery Center, and I recognize Ed Brunikowski here. He's been a great champion, along with Matt Evans, our from the Reptile Discovery Center. They have really been the people with the know-how. They know how to care for animals in our collection. And this is it's an art, it's not a science. You have to know and understand your animals and get them to reproduce. And these guys have been magic. This is Matt in one of our amphibian rescue pods in Panama. We have 2,000 adult frogs breeding in our collection in Panama, and most of them have disappeared in the wild. So here we have Nate Weisenbeck. He has been leading the radio tracking of these frogs. What we did is we basically built little artificial tree holes. They like to breed in tree holes, and mom likes to feed unfertilized eggs to her tadpoles. So this animal got habituated to these little artificial tree holes. We built a little mesocosm around the artificial tree holes so she could get acclimated to the new habitat. And then eventually we just opened up the front door of that and let them out, and we radio tracked them. So they have done very well, and using our passive acoustic monitoring, we have been able to restore the sound of this frog to the forest, which is a really incredible moment for us. Our third release trial was lemur leaf frogs. These have been doing pretty well in captivity, beautiful frog, and they have lots of tadpoles. And so what we did is we decided to get these big cattle tubs and stick them in the rainforest and release some tadpoles into the cattle tubs. And we put a little antifungal solution because they disappeared because of the amphibian chytrid fungus. So in half of the cattle tubs, we put some antifungal solution. And the other half, we left them untreated. But what we have seen is 95% survivorship of these tadpoles with and without the antifungal treatment. So they've been doing great. And when these guys come out of the water, they have these little frogs on the side here. And then there's one of our little metamorphs being released into the wild. Very rewarding, this work so far. And the final species is the Panamanian golden frog. This species went extinct in the wild before I started at the National Zoo in 2009. So I've never had the honor of seeing this species in its native habitat. Here's our project director, Roberto Ibanez. releasing some golden frogs into their kiddie play tents. And they have been doing pretty good. And this is a very special moment for me because it's the first time that I got to see a Panamanian golden frog in the place where it's supposed to be, not inside a glass box. So that is just a magical moment for me. And I had this picture in my mind's eye for 15 years, and I finally got to see it last week. So thanks very much to our team who has been working really hard on rearing these animals and preparing them for release and working on all of the different ideas, all the crazy ideas we worked on the last 15 years trying to solve the chytrid problem. We're just trying everything we can with all the resources we have, and we're just hoping that it'll work. So stay tuned.
Hi, everyone. I'm Kenton Kerns. I'm a curator at the zoo, and I'm honored today to talk about the gold standard, NZCBI's Golden Lion Tamarin legacy. Golden lion tamarins are a small primate found only in Brazil. They're endemic to the Atlantic coastal forest. On this map, you can see the yellow, where the range of the Atlantic coastal forest used to be. Green is all that's left today. Only 7% of that habitat is left. Only 2% is available for golden lion tamarins. As recently as the 1980s, we believe there were as few as 200 golden lion tamarins in the wild. Brazilian scientists sounded the alarm for help, and our own Deborah Kleiman responded. I'm so glad that we got to talk about her so much already today. Because of her knowledge, her skill, her wit, her charisma, dare I say her stubbornness, her inability to take no for an answer, she started an international collaboration, a cooperative management agreement for breeding, really a program that was an archetype for breeding programs into the future that worked with facilities around the world to conserve golden lion tamarins. So through her work, we started to have a boom of golden lion tamarins because we were transferring them around the world. We were collaborating with our in-situ partners to understand how they were breeding in the wild, how to get them to breed better in captivity, to the point that we had huge family trees, these pedigrees of golden lion tamarins. We need to be able to track these pedigrees so that we can have better understanding of their genetics and so that we can avoid inbreeding. We needed to computerize them. This is the very first component of the Golden Lion Tamarin stud book. So how do we get these into computers? Well, our own John Ballou creates the first computerized stud book. He also comes up with a mathematical theorem. When I present this, I think that's insane. Like, I can't imagine coming up with my own theorem for math, but here's John Ballou creating mean kinship, which is something that we're all using in our conservation program. So before we have the ability to get genetic tissue and determine relatedness, we are looking at who is related to who in this pedigree and coming up with numerical values for each animal so that we can tell who's not related to who needs to breed, who's a founder, who needs to be more represented and make these really important genetic pairs. So now we have Deborah's program, we have John's genetic stability. We've got tamarins everywhere. They're ready to go be reintroduced back into Brazil. You can see the NZP logo on the crates as we loaded them into the planes. Our own Ben Beck then is in Brazil, and he's leading the reintroduction effort as we put tamarins back in the wild. So he's managing this reintroduction effort and employing adaptive management. So we're tracking tamarins, they've got radio collars on them, and we are looking at their successes, we're looking at their obstacles, and we're changing our strategies in the field to make sure that they can continue to succeed. And they do. So you can see in these graphs, the red is our introduced animals into Brazil, coming from 43 different facilities, 153 animals over about 20 years. Blue is their descendants. We had 1970, in the 1970s, we had 200. We're now over 5,000 tamarins in the wild today. We believe that 50% of the wild GLTs are descended from zoo-born golden lion tamarins, and we are almost certain that if we had not reintroduced those tamarins, this species would be extinct. So where do we go from here? Our own Jennifer Mickelberg came up with, during her PhD research here at SCBI, she came up with our science-based standard, which is at least 2,000 GLTs, which we have, living in about 25,000 hectares of land that's connected and protected. That is our goal. That's when we can take a step back and know that we've done right by the tamarins. The good news is we know what we're doing. We know how to connect forest. This is a typical forest fragment in Brazil that tamarins cannot cross. 2010, 2017, because we know what we're doing. Another piece of land that we own, 2018, 2022. The first forested overpass ever created for wildlife in Brazil was created for golden lion tamarins. We're getting there. We've still got a long ways to go, but we're close. 5 minutes is not enough time to talk about the history of tamarins, what we're dealing with right now and what we're dealing with in the future. But my hope is that we can all look at this and understand how pivotal all these people have been in getting us this far. And if you want to help, please come find me. Happy to talk about tamarins. Can talk about it for hours, but 5 minutes was all I got. Thank you.
Hi, I'm Chris, here to talk to you about creating cranes through AI. We are the leading producer of white-naped cranes. We've hatched 49 chicks here, 21 of the last 25 through AI. AI is done by staff here, by keepers. We catch the birds in their enclosure, hold them in a standing position, and 2 people manually massage them in the relevant areas to get the response. AI is necessary when cranes can't breed naturally because of behavioral or physical issues. The main issue we see is behavioral, where decades ago, other zoos were hand-raising cranes, so the cranes imprinted on people instead of cranes. These mal-imprinted cranes typically don't get along with other cranes, and even if they do, they're not able to breed naturally. So we get sent the most genetically valuable ones in human care, most genetically valuable because they haven't reproduced to pass their genes on in the captive population, which is an insurance population in the event that the wild population continues to decline. And so here are some of the success stories. None of these chicks would have existed without the AI. So this is the pairs that we bred through AI and the chicks produced. We've also done AI with endangered hooded cranes and endangered red-crowned cranes. We're trying it with endangered whooping cranes. They're the rarest crane in the world. They're native to North America. There's a lot of issues with the ones in human care. We have 8 individuals, only 2 can breed naturally. And any chicks produced here can be part of a reintroduction effort in Wisconsin. So the most famous success story is Walnut. She should have been reproducing at 2 to 3 years of age, but she came to us at age 45, never having produced any chicks because she was hand-raised by people at another zoo. She's the most severely imprinted bird we've ever seen. Other zoos tried to pair her, and she allegedly killed 2 males that they put her in with. I say allegedly, it's not like she was tried and convicted. So she was sent to us so we could keep her separate. We could do AI, and we could give her put her fertile eggs in the nest of another crane pair so they would be raised as they should be by other cranes. So we got 8 chicks from her. The birds on the right are Brenda and Eddie. Brenda is the middle bird. That's Walnut's first chick. So she having been raised by cranes, she's perfectly normal. And Brenda and Eddie have had 5 chicks all on their own. And they're such good parents. They're also been surrogates for some of Walnut's other chicks. And because Walnut was imprinted on people, and because I took care of her every day, we developed a pretty strong bond, and we thought that I could try doing AI without any restraint, just going in myself, keeping in mind she had killed the last 2 guys that tried that. So over many months, she was able to train me to do AI without restraint, which is pretty cool. So this is a video of her training me. So she's walking away, but she still has allowing me much closer than other cranes. I got her to come close to hand feed her some food. I'm gonna need to touch her lower back to do the AI. She moved away, but not far. So I'm gonna try again. Moves away again, but she's doing a little courtship, a little head bob. And then I'm gonna toss her some nesting material. That's also courtship. I'm gonna need to touch her inner thighs and below her cloaca. And this was a breakthrough where she just stood there, allowing me to touch her. And here she is dancing, and I would, flap my arms up and down. Here she is soliciting me to mate with her. Most people would be arrested for this behavior. But I'm massaging her lower back, her inner thighs, and around her cloaca. I have a syringe with the semen sample that once she's receptive will just be inserted into her cloaca to make a baby crane. And if you'd like to know more about Walnut, you can buy this family-friendly book from SI stores or wherever you buy your books. Thank you.
I think that's going to be very hard to follow that talk. But oh well, I'll try. So good afternoon, everyone. I'm Nucharin Songsasen. I'm the research biologist and the head of Center for Species Survival. And today I'm going to talk to you about the love of my life, the Maine wolf, of course. But before I do that, I would like to thank Colleen for organizing this session and allow me to blabbing here for 5 minutes. And thank you so much. Let's begin. All right, the Maine wolf is one of the very first species that we have here in this facility, even before this facility become official. So they arrived in 1974. And in this picture, you can see that Dr. Reed holding the Maine wolf pup that actually born in this facility. This is in 1983. I have potential name for them because the sorrel pups are born that year, so I'm not really sure which one this is, but as you probably agree with me, they're pretty cute. And since then, NZCBI, animal care, veterinarians, and scientists are the leader in research and conservation of Maine wolf. Everything that we know about Maine wolf, most of it come from this facility. Staff in this facility and also at the zoo generate knowledge that we know about this species today. And some of these pictures, you know that some of you recognize, some of them are in this audience. And then those are some students that we have trained throughout the year, and some of them have become the leader in the field as well. So the research that conduct in this facility had a link between XC2, that means that animal in under human care and also in the wild. In addition to conducting research, we also are the largest facility that house Maine wolf. Now, until today, we have about 143 individuals that we house here in this facility, and 4 of which are the wild-born that we brought in from Brazil in early year. And we have produced nearly 100 pups so far, and we continue to allow to breeding this species. Will be over 100 pups that born in this facility. So NZCBI staff had played a role in program leader. So we have species coordinator for Maine wolf species survival plan, Melissa Rodden, the first one. And I'm also the coordinator of Maine wolf species survival plan as well, as you see how much I love Maine wolf. So we also conduct research in all sorts of disciplines, starting from Dr. Deborah Kleiman, start working on social behavior of this species, and learn that they are solitary species. That means that male and female do not spend time together. They only got together during the, for a date. And then we're looking at veterinary medicine, husbandry, reproductions, ecology, genomics, everything you name it under the sky. Staff here has done it. So far, we have published at least 53 publications. I did Google Scholar. This is all I found, but I'm sure that there are some that I probably missed, and 13 theses from graduate students. These are some examples of those publications that we have published until this year. And why is it matter to have species here at SCBI? So we are as I said, the largest facility that house Maine wolf and the leader of the Maine wolf SSP in the ACA population. What we do is important for the SSP and how this animal in ACA in North America can support those in Brazil. In Brazil, zoo receive Maine wolves every day from the wild because their habitats are gone. However, they don't have enough space to conduct the breeding themselves. So none of these wolves are bred. They just sit there and lost the genetic diversity that otherwise could contribute to the entire population. So by having population here in the US healthy and more facility supported, then we can bring those individuals from Brazil, conserve those genetic, and then the zoos in Brazil can also breed the wolf because now they have space to breed wolves and maintain that gene diversity that we need. So having the ACA population also play a role in the wild as well. And by having these animals here in this facility, allow us to conduct a lot of research, filling knowledge gap that otherwise would not be able to do. And then research or knowledge that we gain can turn into tools that can then be applied to the wild population. Such as we develop non-invasive hormone monitoring, so we can now looking at hormone concentration of wolf in the wild, that we have published a paper. And the most recently that we have developed using the biologger technologies to look to understand the health and well-being of animal, both in under human care, also those in the wild. So what next then? I still have not given up on Maine wolf yet, and hope that none of you will be. So we just recently received a grant from ACA to optimize sperm cryopreservation for the species. This is very species that very hard, their sperm very hard to freeze. And I bang my head in the wall every day. How could I do that? So we got funding to do that, and hopefully we can optimize these techniques and use it to bank sperm of wolf that in the US and also in Brazil and even in the wild. And then we also have ACA grant pending on developing biomarker for disease discovery or to looking at the health of individual and the populations. And I hope that the knowledge that we accumulate with Maine wolf can put into digital biobank that Molly and Buddha has talked about as well. So gonna be collection of multiple species in this digital biobank. So we collaborate with our Brazilian colleagues in this hologenomics, which is, this beyond my head, but it's about combining genomic, microbiome, epigenetics, everything that you know about genetic of the Maine wolf. First start in Brazil, and then eventually will be in the US and in globally. And we still develop in the idea of how we can non-invasively detect disease exposure. With my conversation with colleagues from Kenton specialist group of the IUCN, it will be good for us to know whether there's animal in the wild have been exposed to disease, to understand disease epidemics in the wild. So by having animal here, I said, well, we vaccinate our wolves all the time. So perhaps we can detect some of those antibodies or some sort of indicator that the animal has been exposed to disease by using collection that we have. I also toiling with the idea of biofence. So you may wonder what biofence is. Biofence is the way that you can use compounds from either feces, sand mark, urine, or any compound that's similar to what animal excrete to spray around the farmland, and that prevent that predator to go into the farm and cause a conflict. And this also can help reduce the human-wolf conflict in Brazil, because this is one of the main threats to the species. And we would like to continue to be leadership in ACA. We has been a leader, and we will continue to be a leader. And with all of this cannot be done without support from all wonderful donors and foundations and granting agencies. So I would like to thank all the supporter, just not for me, but for the whole program. And thank you very much for your attention.
Good afternoon. My name is DJ Washington. I'm a bird keeper here at SCBI, and I work with our loggerhead shrike. So I'm going to tell you a little bit about our program with this species, our history, and our success over the years. So without further ado, let's get into it. So your first question might be, what even is a loggerhead shrike? To kind of give you a brief overview, they are a songbird, but unlike your typical songbird, they have an ecological niche that's closer to that of raptors. But unlike raptors, they have a very unique impaling method when it comes to dispatching their prey. So they will use thorns, barbed wire, any kind of sharp edge that they can find in their environment, and that has earned them the nickname Butcherbird. So the loggerhead shrike is the only shrike species exclusive to North America. In fact, it's a species that can be found locally here in Virginia. And here at SCBI, we work with the eastern loggerhead shrike, which is recognized as a unique migratory population. So these birds breed in Canada in the spring and summer, and then they overwinter in the eastern US. So why work with a species like the loggerhead shrike? Like so many threatened species, they're disappearing and disappearing fast, as much as 76% across their entire range. In fact, the eastern loggerhead shrike is considered critically endangered in Canada. And that decline is strongly associated to a multitude of factors that are listed here. So where do we fit into this picture? The Eastern Loggerhead Shrike Recovery Program is led by our partners, Wildlife Preservation Canada, or WPC, and the Smithsonian joined in 2011 as part of its breeding initiative and reintroduction program. So since 2011, SCBI has produced over 264 loggerhead shrike chicks. 200 of those have been released into the wild. I'd also like to highlight that 2025 was a really big year for us. We had 44 chicks produced, the highest that has ever been produced to date. And those 44 chicks accounted for about 72% of the releases into the wild this year. So that's really amazing. And you can see on this graph here just how drastic that improvement has been from where we started and where we are now. So obviously this breeding program is helping to ensure that we have a genetically robust and healthy population of these animals in human care. But as I've mentioned, we're also directly supporting the stabilization and increase of the wild population. And we also participate in a multitude of research studies ranging from migratory behavior, the collection of blood and feather samples that can be used for gene and population analyses, and even trialing remote tag software that can be used for long-term monitoring once these birds are in the wild. I'd also like to highlight two really amazing instances we had this year in Maryland and Virginia, where we had local birders identify loggerhead shrike, which is exciting in and of itself. But what's also really special about it was that these birds were banded, and it was able to be identified that they came from SCBI and had been released the year prior. So when we take all of that into a bigger picture, we're not just breeding birds, but we're also helping to address those knowledge gaps in our understanding of the species. We're helping to engage the public in the recognition of the loggerhead shrike and its conservation concern, and we're shaping new management practices in both the in situ and ex situ populations. So where do we go from here? In just a little over 10 years since this program has started, we've made monumental progress and huge contributions to the recovery program at large. SCBI has quickly become one of the most productive breeding facilities for loggerhead shrike out there. And I think that really speaks to our expertise and skill set across the board here at SCBI and our ability to not only just bring species back from the brink, but return them to the wild. And the most exciting aspect of all of this is that this growth is continued to increase and go up. If this is what we can accomplish in 10 years, imagine where we can be in another 10, another 20, or even 50 years from now. It's really exciting to think about. If there's anything to take away from this, it's that SCBI has and will continue to be a monumental pillar in ensuring a future for North America's only butcherbird.
Hey, everybody, me again. So now I want to talk to you a little bit about our black-footed ferret program. And again, first, a little bit of history. So black-footed ferrets are the only endemic ferrets in North America. Their range once spread from southern Canada through the Great Plains states and down into northern Mexico. Unfortunately, in the 1970s, black-footed ferrets were thought to be completely extinct in the wild. And this is due kind of to the usual suspects, loss of prey and loss of habitat. So in the early 1900s, farmers were actively changing the Great Plains into farmland. And as part of this, they were actively trying to eradicate the prairie dogs from the land. Prairie dogs, however, are the black-footed ferrets' primary food source and also provide their housing. So the black-footed ferrets live in the prairie dog burrows, which is great access to their food source, but also along with this comes the disease-carrying fleas that live in the burrows with the prairie dogs. However, a last population of black-footed ferrets was discovered in Meeteetse, Wyoming in 1981 by this dog, Shep, brought a dead black-footed ferret home to its owner. It was determined to be, in fact, a black-footed ferret. The population in Meeteetse peaked at around 129 individuals in 1984, but then unfortunately, shortly after that, the population started to crash due to canine distemper virus and also sylvatic plague, which is carried by those fleas. In 1985, Fish and Wildlife Service started the process of removing ferrets from the wild to bring them into human care to start a breeding colony. 24 animals were brought into breeding centers, and this began the Black-footed Ferret Recovery Plan. So this was definitely small population management with only 24 individuals. But SCBI was one of the first breeding centers for the species, and researchers here were among the first to document high levels of abnormal sperm in the species and did a lot of other reproductive research trying to generate as many black-footed ferret offspring as quickly as possible. Again, Dr. Jogel Howard was the first to develop artificial insemination in the species, and it is a very successful technique resulting in over 130 kits produced. This program is so successful because of the vast number of partnerships. We also have folks based in Montana as part of the Great Plains Science Program. This is Jesse Bulleries. He is based in Montana, and he's doing lots of different projects to investigate how we can improve the survival of the black-footed ferrets after reintroduction. So he's using these fine-scale trackers on both prairie dogs and black-footed ferrets to try to better understand their movement ecology, and even using drones and AI technology to try to understand their movement behavior below ground in those burrows. We collaborate with a lot of folks on the ground that are actively trying to reduce the fleas in the prairie dog burrows. So we help support the plague mitigation. You can see in this picture, there are people that drive across the prairie and literally spray dust in these prairie dog burrows to try to decrease the fleas that are living down there. So our success has been pretty noteworthy. We marked our 1200th kit produced this year, which is exciting. So since 1989, 1200 kits. Most have been produced by natural breeding, but again, over 130 produced by artificial insemination, which is very successful for our carnivore species. So every year we produce as many kits as possible to go back out into the wild at these release sites, and also to help support the XT2 breeding program. And most recently, we have been involved in conservation cloning for the species. So if you remember those 24 individuals that came in the 1980s, scientists cryopreserved cells from one of those individuals. She was studbook number 10. Her name was Willa. Recently, scientists have been able to make embryos from those cells, so a clone embryo from Willa, and produced offspring. We have one of the offspring here. Her name is Antonia. She was born in 2023. And importantly, Antonia has produced her own kits, a litter each in 2024 and this year. Antonia represents the first endangered species clone that has contributed offspring to her population, to her species. So these genes that were from the 1980s have now been reinvigorated into our contemporary population, helping the gene diversity of the XCG breeding program.
All right. Good afternoon, everyone. I am Mel Songer, and I am the Deputy Director for Conservation and Science. So that's the first time I got to say that. So thank you. It's an honor, and it's a big responsibility too to follow up this fantastic series of talks. So thank you all for joining us today. I think we've seen many examples of breakthroughs and conservation happening in real time. I'm confident that there's no other organization that has as much experience in the critical disciplines that are necessary to restore species in the wild. And this expertise goes from the zoo to SCBI, from animal care to veterinarians, scientists, technicians, and so on. I think you can really see that today. It's a major strength that we have, and I think it would be extremely powerful to think and speak about this more effectively. And so this is a term that Peter and I have been kicking around, WildFutures. I think this could be something that would help with us communicate better among each other and also with the outside. We're really able to take a holistic approach from saving gametes and diversity, husbandry and care to maximize welfare, sometimes even preparing animals for release. Growing populations through advances in artificial reproductive technologies, monitoring animals in the wild to enhance their chances for survival, and even using genetics and genomics to assess disease and impacts of environmental change and population status. And ultimately, whatever we're learning, we're sharing it with the world. I think we have the potential to be kind of a one-stop shop for figuring out what it takes to restore a species. So today, I just want to give a couple of examples how we are overcoming challenges of species reintroduction, and especially how we're using science and partnerships. But in these examples, it's especially important that we had input and assistance from animal care staff that wouldn't be possible without them. So we know restoring a species, it can help bolster wild populations, can stop cascading effects of extinction, but only 1 in 3 reintroduction projects is successful in the long term. And often monitoring of these projects only happens in about a quarter of the times where we're actually monitoring and figuring out whether it's a success. So post-release monitoring is really critical for advancing the science of reintroduction and also improving chances for success. Movement tracking, of course, is the most powerful tool for monitoring reintroductions and learning about a species ecology to inform adaptive strategies. We can find out about their movement, dispersal, reproduction, behavior, how well they're adapting, and social dynamics, but most importantly, whether they're surviving, and if not, why. We're leading experts in post-release monitoring, as well as working with local partners and stakeholders to inform management and adapt for success. Our Movement of Life network of partners has collected over 2 million points from, and this is an old figure, but even more than that now. And thanks to Autumn Lynn, who calculated this number, 111 birds, mammals, turtles, frogs that we have had a part of tracking. And that includes 20 reintroduction projects. So we're not only tracking with traditional methods, but also innovating ways to develop lower impact, longer lasting, and lighter ways to track animals on land, air, water, and as Adrienne just mentioned, even below ground. And I want to talk a little bit more about that. Adrienne helpfully queued this up for us. And, you know, this has been a really important breakthrough in terms of having this keystone species and being able to figure out what's happening underground. Even though we have done major work in terms of developing the reproductive technologies and producing over 1,200 kits, been providing them for reintroductions. Even after decades, there are still challenges because of their reliance on prairie dogs and the fact that these towns are regularly plaguing out. So we really need to understand better what's happening underground, and this can be very challenging. So as mentioned, Jesse has been working on using cutting edge technologies to be able to see what's going on after they're under the ground. And this is the design for this, if you think about, is very anatomically challenging. So no offense, Adrienne, but ferrets are essentially a furry tube with arms. So it'd be very difficult to keep a collar on them. And this is work that started here, trying to develop a good collar that would work. So to track the fine scale movements, they put together with partners high resolution sensors containing accelerometers and magnets, basically a dead reckoning system that continuously tracks data, even 40 positions per second, and to be able to then apply trigonometry and map these three-dimensional paths. And so once we have those paths, now we can actually see the shape of the burrows underground. Here on the left, you can see, which is something we've never had before, even being able to see potential nest spots. And we also can learn about their behavior. If you look on the raw data on the right-hand side, you can see the resting periods, where this is likely places where they're stopping and potentially nesting, and then these rapid changes, which in the ferret's case, probably hunting, and potentially for prairie dogs, they are running away from predators. So this has really been a huge jump forward in terms of recovering these keystone species, learning about their interpopulation dynamics, disease ecology, predator-prey relationships, and recovering these keystone species in our Greater Plains grassland system. So hang on, I'm going to make a big jump to the other side of the world for one more example. Working on working elephants in Asia. And this, no pun, but pun intended, I should say, this is a huge species for us here. We have been involved even from the beginning in terms of developing reproduction, being able to build these populations, healthy populations under human care, but also in the genetics and genomics and the welfare, discovering the EEHV. So we have really been critical in terms of building these populations in zoos and breeding centers. I'm going to focus on rewilding. This is work that we're doing more recently. There are over 30,000 work elephants, and demand for them, however, is going down in some countries. So we've had the opportunity to begin to work on how we might be able to release some of these working elephants into the wild. The challenge is that these elephants are actually very familiar with people. So there's a high risk of engaging in human-elephant conflict. So that's something we want to try to figure out. Are there elephants that potentially are going to be more likely to succeed? They're going to be less likely to cause conflict. And so this really began at the National Zoo, doing training or doing testing to see on the behavior, to see how they are reacting to novel things like a puzzle feeder, if they're neophobic, if they're afraid, or whether they're engaging with a puzzle feeder, and what kind of exploratory behaviors they're engaging in. And so what we found is that there are very big differences in individuals. So we are seeing elephants that are more afraid and also are less exploratory. We think that those elephants are going to be the ones that do better if they're released, because hopefully they will stay put and not wander off and get into trouble. So another focus has been on their sociality, because when these elephants are released, potentially they're going into places that already have elephants. So working in elephant camps, been able to do tracking of individuals. So if you look at the map here, the 4 different colors are 4 different individual elephants. And you can see how green and yellow kind of like to be on their own compared to orange and red, who play better with others. We think they are more likely to succeed and do well if they're released into an environment with other elephants. So this is work that we are continuing with the Elephant Conservation Center in Laos. And this is a place where actually Janine's been working for many years, building an endocrinology lab and technology and capacity there. Last year, Michael Campana began setting up a genetics lab and training the capacity for them to be able to analyze DNA samples. And this will really be important for obtaining baseline data about their demographics in the wild. So this is Laos has few elephants and not a lot of places, but not a lot is known. So this will be really important in getting the staff trained up, getting samples, and figuring out what's going on with the population there. So ultimately, what we learn will inform future releases and will help with the welfare crisis that can come with unemployed elephants, supporting restoration of healthy working and wild populations throughout the range. Oops. So just one more challenge I want to mention. A lot of times when these reintroductions happen, it's yay, we did it, and there's not so much follow-up. But it's really critical that we continue to engage, to adapt, improve, address new challenges, working with local stakeholders and partners in order to be successful in the long term. And this is another thing that I think we really do well as an organization. So I'll just, in the way I started, I'm confident there's no other organization that has as much experience in the disciplines critical to species restoration. We really do it all across disciplines and departments. I do think there's more we can do. Sometimes I think there's a disconnect where we don't have a mechanism for communicating. And I think this is something we could do a lot better on, connecting curators, animal care staff, veterinarian scientists. And this is something I'm going to be looking for ways to better communicate with each other, to message to outside partners. I just want to point out what you've seen today. I think it's coming clear how committed our staff is. We have staff that are willing to date cranes and, put a stuffed badger on a remote controlled car. And, ultimately working hard, figuring out what it takes and the commitment and dedication to figure out how to get species back in the wild. And I just want to say I'm so proud to be here, and I'm so proud to work with all of you, and just so looking forward to the future and what we can do together. So thank you. So I just want to say thank you to Colleen for organizing that session, and to the speakers, just fantastic job.